Hello? Yeah. Okay. I hope everyone is not tired already. But first, before I start with my talk, I want to. Oh shoot. I want to say I come from academia normally and cancer research, and when I go there for conferences, that is so much different here. That is incredible. So inspiring people and inspiring talks. That is, yeah. So I hope I can contribute to that. So I want to talk today a little bit about space biology and why is that actually important for our endeavor to become a spacefarer or a space settler, or another title for that, why spaceships are just not enough for that. And we live in really exciting times. We saw that the whole day and yesterday, that we are actually on the brink to become a multi-celestial species. And this time is almost that important, I guess, like our ancestors when they sailed over the oceans and occupied new islands or new continents and brought actually their plants and their culture with them. But what they had to do was, of course, they had to adapt to the new environment. We saw that in the talk about Alaskan people. What was really interesting, actually, and fits totally into that. And but our frontier is, of course, very different because we have a very different environment, and our ancestors had similar environments, like temperature range is okay. That is has a variety, but the air pressure is the same, and the radiation and the gravity is the same. So while we actually know, oh, yeah, that's very sensitive. Okay. Okay, while we actually know how we get to Mars or to other celestial bodies, or how or who brings us there, and how we live there, maybe that is a lunar base I picked up from Google somewhere, so I'm not sure if that really will look like that, or on a Mars colony. So we actually need to adapt ourselves and our companions, what we bring with us, our food sources and bacteria and everything, to the new environment. So, and that is actually a field, what is the space biology about? So, space biology is how organisms from Earth, like, I mean, we don't know other organisms right now, would behave in space-related environments without gravity or reduced gravity or higher radiation. So I want to start a little bit about this, about us, how we behave in space. And besides all these stuff, what we already know, that we have a problem with the muscles and microgravity and with the bones and with the immune system and the heart and the blood pressure and the immune system I told already, yeah. So there are other things, and NASA actually did an experiment what was quite interesting, and I'm sure you know already about what I'm talking about, but that was about the long-term space mission of Scott Kelly. So he was 1 year in space, and the amazing thing about this study was actually that it was a twin study. So Scott Kelly was in space and Mark Kelly was on Earth, and both had given samples like blood and other samples, and the scientists studied that. So he was 1 year in space, and when he returned, they started to have to look up the results. And the sad thing is, right now, to this date, he landed last year in February, I think. And this year in January, they had a little article on nature.com about the preliminary results, but there's no paper out right now. But I want to talk about these preliminary results, and this is actually, so the first one is rather boring actually, but they said the gene expression in space is different. Okay, that doesn't say anything much because they didn't say if that is higher or lower or even what genes, but the next experiment or the next result is quite interesting actually. Scott Kelly in space had a change in the telomere length. They got actually longer. And since I'm here on a more with physicist filled audience, I want to explain a little bit about the telomeres. Telomeres are the ends of the chromosome, and they are heavily involved into the aging process. So after each cell division, the telomeres get shorter and shorter, and older persons have shorter telomeres than younger persons. And there are some theories out there when you can crack the code and extend the telomeres, and you can live longer or even forever. So these are some ideas, but in general, it's an aging process. Telomeres get shorter while you age. And Scott Kelly's telomeres got longer in space. And when he went back to Earth, the telomeres returned to their length before the space mission quite in a few weeks. So that is quite a astonishing finding, and NASA actually has no clue why that is, and they launch another experiment, and the result will be in 2018. So I'm very curious about that. And since I'm in epigenesis, I'm very interested about the third result. So they found actually an overall change in the methylation level in the genome. And since that is more a biological topic, I also explain that. We have 4 bases: adenine, cytosine, guanine, and thymine. So, and in the context of cytosine and guanine, the cytosine can be methylated. Yeah. So that is a process. I don't want to go into that right now with enzymes involved and so on. So it can be methylated. And when this cytosine is methylated and appears very often in so-called CPG islands, CPG islands, which appear normally in front of the promoter region of genes, and the promoter region is responsible for the activity of genes. So when is a gene active, how much is a gene active? The CPG island, if the CPG island is demethylated, the gene is active and is expressed. When the CPG island is methylated in the promoter region, the gene is actually inactive and the gene expression is silenced. So, and when we now say that his overall genome methylation is decreased actually, then it means that a lot of genes are active, or more genes are active than normal should be. And to illustrate that, how complicated that is, imagine any epigenetic or any genetic program as a song on a piano, and each key represents a gene. And when you push down the key and you hear the sound, the gene is active, and you have to, in order to play a song, you have to push the keys in a certain order and length and duration, and so then you have the song. And when you have a lack in regulation of the CPG islands because of the lack of methylation, then it's like you just push all keys down at the same time. So that is very, that illustrates that there are more problems going to space right now than we know already. So that is, we need to do much more research in that case. So how to overcome that, or how to prevent that, or what is also very important, how much G-forces a human being would need to overcome this development. So for plants, for example, the threshold 0.3 G, and over this threshold, plants can grow quite normal, and under that, they have problems to grow in microgravity. So we need to figure out what this threshold for humans is. And now I want to talk a little bit our companions, what we have to bring with us. So, Angelo talked about that already with Melissa a little bit. So what we would need, that would be an oxygen regeneration system, a food source, and a recycle system with bacteria. And here we see, we need to bring several food sources, and here this is cyanobacteria. Cyanobacteria have the other advantage that you can produce biofuel out of that. So you may ask why you need biofuel on Mars, but there's an idea out there, since Mars gets only 50% of the sunlight, solar panels wouldn't be a great idea, actually. I mean, you have solar panels, but that cannot be the only energy source. So you would produce actually biofuel with cyanobacteria and would burn them in normal combustion engines with, not normal, because you need to provide also the oxygen, of course. So that is one idea. And for this, one second. But since cyanobacteria also would need light, you have only 50% of the light source from the sun, you would also again need artificial light, and that would negate the whole program. So I want to propose today another organism what fits much better the needs for biofuel production on Mars without light, actually. So what you see here is maybe a new space cadet. That is a slime mold, Physarum polycephalum, and what you see here is one single cell with no cell wall, just a plasma membrane. And this organism has different advantages. There's one advantage. It has been shown that this organism has actually the very good combination out of lipids for the production of biodiesel. And another advantage is it can grow on very easy carbon sources like organic waste, what we would have from the plants' rest, what we would left over, what we have. So, and there are another ones. So it's an ideal one. It can also be harvested much easier than cyanobacteria because they don't have a cell wall, what is much less energy extensive, and they grow without light. They can grow on the material, and they are actually radiation resistant. I tested that actually myself with gamma radiation when I was back in LA, and they don't care about radiation. So that is very interesting. So what I want to say with all these talks is actually, with all these results, that we need to look out here on Earth what we can use for organisms and for biological systems to adapt ourselves and our companions to the space environment, because just getting there is not enough. We need actually to adapt ourselves to get a true spacefarer. So, and that is my talk. Thank you very much.
Excellent stuff. Questions for Christoph? Angelo?
Well, I have a lot of questions actually. First of all, that was fascinating and such a brief talk. An astronaut was sent in space, and his life was extended. And when he returned back to Earth, he recovered his original age. So you go up to space, you don't basically want to come back. Isn't that kind of it? Second, go ahead. Did we take it?
So my theory about the telomere length is actually there's a life extension diet, a low calorie diet, where when you feed apes and monkeys with low calories, they look actually much younger, and they behave younger when they are older compared to a heavy diet. So my idea is that the body wants to overcome this kind of starvation stress to reproduce after that. So just hold off. We cannot die. We need to produce. So maybe the idea about the space habitation is the body has a lot of stress. There's even stress no organism on Earth actually experienced since life emerged on Earth, right? So, and the idea is maybe that the body has some programs going on to extend the life, or to extend the life to a certain degree, of course, to overcome that stress, or wait until the stress is over. But that is only my idea, so I'm not sure about that.
But again, the second conclusion was, the other thing that you showed was that the genes were, otherwise latent genes were being expressed. Yes. And the combination of those is, so first of all, that gave me the idea that perhaps the body is looking for adaptations to its new environment. It's detected a new environment, the high stress. It's trying to express other genes to see if it has some kind of competitive advantage in the new environment. So space and or other environments are drivers for evolution biologically, so we're triggered. A switch goes off, goes on. That is actually and it extends your lifetime to find enough time to adapt.
Yeah, that is actually a very interesting idea, that the body tries to adapt to that, and that is all what epigenetics is about. Epigenetics is actually the software of the DNA. When you go somewhere, or when you even eat other foods, your epigenetics changes. Or when you smoke, that is the reason of microinflammation. But when you do stuff, and the environment changes, your epigenetics changes too, and that is the whole thing. If really all genes are expressed, that is a big question. I don't think so, because then he couldn't extend his life. But I don't know what genes. That is a big question. I mean, we need to wait for the article. And I visited 1 month ago actually a talk in San Diego about that, and they didn't even tell so much like here, and it was already public, and they said, yeah, in a month or maybe in 6 months, so we get the publication out. So I'm very curious about that. But it has actually a deeper level. When you think about that, epigenetics is very crucial in the development phase, even in the development of the eggs and the sperms. It's very, very crucial, and also in the first phase of the development, the whole embryo has to demethylate it first completely, and then methylate it depending on the gender. So you have gender-depending imprints of the genes, and when you have a problem with that, sustain that in space, then there will no offspring. That is very. Also we need. For spaceships, that is not a problem, because we can have this artificial gravity. But for a moon base, with the gravity of 0.16 or so, I think is that it's very, very low. Mars, we don't know. We need to figure out what is the threshold where a human being has no effect on or microgravity has no effect on human being.
I have a question about the slime mold. I've been working with slime mold as well. Have you considered in your research the computational qualities of slime mold? For example, I used it with a research group in Vienna to optimize the pathways between buildings, for example, and we scanned the results of the slime mold connecting small structures, and then we use that to design architecture. But there are many other examples.
Yeah, no, the thing is, I found this organism a couple of years ago, I think 2011 or so, and I was so fascinated by that I played actually in my free time in my lab, not my lab, but I had other stuff to do, and so I didn't do really much research. I did some atmospheric research where if they grow under different or with an oxygen-less atmosphere, so I built some stuff there, and then I irradiated that, and then I did also some epigenetics tests, and actually this slime mold has epigenetics to a certain point. Actually, the epigenetics is different in the, when I go back to that, the epigenetics is different from this part here and to this part here. So there is a different methylation going on. Or what I didn't tell, that is one cell, but there are thousands of nuclei inside.
Right?
So yeah.
The cell walls are gone, basically.
Yeah,
right?
cell walls are gone. Yeah.
Like
so that is.
But yeah, the work of, for example, Andy Adamatsky, he did wonderful research. On
I have his book actually. Yeah.
It's just really. The second question: when you encounter these organisms in nature, they're usually relatively solitary. Have you envisioned how you would mass produce this? Because with algae, it's pretty obvious. You go to a pool and you see all these algae, and it's.
Yeah,
but in a forest, I've never seen like a mass production of.
No,
of slime mold.
it's right. No, you can produce them in a bioreactor with a spin, and then you have microplasmodia. So they actually the whole thing gets not destroyed, because it's like you can fuse them and separate them as you wish, and you have little droplets of the slime mold, and then it grows, and when it's too big, it disintegrates again.
I guess a comparative experiment in mass producing algae, and then this one, the slime mold, and then producing the output and the input. The comparing output-input would be a beautiful
that grows
paper.
actually very fast.
Yeah, it would be a beautiful paper. So
it would be, yeah.
Go for it.
If I could, I wish, yeah.
I have a follow-up question about Scott Kelly. I know the research isn't out yet, but you have a couple of factors that perhaps are influencing the changes that are being observed. Microgravity is one of them.
Yes. Of course, yeah.
Diet, maybe another. But what about homeosis? The astronauts experience 2 orders more magnitude of radiation than a surface dweller. Low-dose radiation usually helps the immune system perform better. How do you establish what the root cause of these expressions when you have this many different factors to look at?
Yeah, since we don't know what genes are actually upregulated or downregulated, so I cannot really tell you something about that. But the methylation, in general, the thing is, I studied that. I studied, or I have a few papers about the acute gamma radiation on the influence of gamma radiation on the epigenome just in cells, and the acute change. There's actually no change in the epigenetics, but in the accumulated in the chronic experiment, we didn't publish that. That is very sad. But we had cells. They are accumulated actually 21 gray. And they were, you couldn't tell they are human anymore. So, of course, but I don't think that Scott Kelly accumulated so much gray, so that's very, very low, and it's a low level. We didn't see any changes in the epigenetic scale, not what they propose here, yeah.
I heard he exercised a lot too. Did he exercise more than astronauts normally do? Could that have something to do with the growth?
Yeah, there are actually several issues with the whole experiment. In the talk I was referring to 1 month ago, they said actually that his whole biome, that the bacteria in his biome shifts. So one strain was very low and the other strain was very high, and then one guy in the audience asked actually, did Mark Kelly on Earth also eat the same thing like Scott Kelly? And they said, no, no, no. And even we are not sure if Scott Kelly ate what he signed in, because there is this, this behavior up there that they trade their food between the different nationalities. So the whole biome thing is very, that could also be a reason, but not on this scale. There's the whole genome decreases in the methylation. That is very weird. And the more exercise, that wouldn't cause that too. I mean, okay, I'm not sure, because the experiment is a little bit flawed, because Mark Kelly on Earth didn't do the same thing exactly. So that's a little bit sad, but yeah.
Yeah, question. First, is that not necessarily algae or slime? Is that consumable by humans?
At least it's not poison. I never tasted that. So
I was just curious whether or not it's consumable. That's
yeah, I think you can eat that. The funny thing is, I have that actually here. That is a life cycle on the slime wood. It's an incredible organism. You have here the plasmodia, what I said, and it can actually dry out and then when you put water on it.
It's like mushrooms, right? It's a spore.
It's not like mushrooms, because they have also a state. They have spores here, yes, but they have here also a phase of kind of swimming amoeba and then amoeboid cells, and they can actually also interchange depending on the environment. And when they fuse, then they start growing. So this organism is almost like alien. That is, yeah. Mm-hmm.
And then the second question I had was, since you were talking about genetics, has any study been done for the offspring of individuals that have been in space and came back down?
That is interesting. I know Mark Kelly retired and he has a wife, so I don't know if they have actually a kid, but yeah, but that is difficult, ethical, right? So, and this, and they talk that also, we know these people. Everyone knows these people. That's why they don't want to release too much information about their genetics. That's why, yeah, maybe there are actually informations they don't release. So it's, yeah.
They are the aliens.
Yeah.
All right, so Chris, another question? Yes, certainly. Because we're using all the time in the world today. Yeah.
Right, you're doing vet plants too. Has any thought gone into starting to hybrid plants so they live in lower gravity or with less light?
They figured out the threshold because of the ISS studies. So they grow plants in space in the ISS, but that's not really effective. And there's a paper out where they estimated the threshold, what is 0.3 G, where below that plants would have problems to grow, and over that they wouldn't have a problem. On Mars is a gravity 0.376, I think, and that wouldn't be a problem, at least for the estimation right now. So otherwise, for the moon, you would need actually to really breed plants that they grow in lower gravity. But for that, you would need to know why the plants don't grow with lower gravity. That's why the whole research is going on. And that is, so far, I think, not really revealed why they don't grow much. Yeah.
All right. Christoph Lahtz.
Yeah, thank you.
Thank you. Thank you very much. 2-minute break.